As Veronica said, my name is Harry. I'm a postdoc at the University of Texas at Austin, and today I'm going to be talking about novel insecticides and how they influence beneficial insects. So, as I'm sure everybody listening to this talk will already know, insects are important for crop production. And today I'm going to be talking about two different groups of insects, pollinators, which in the data sets I'm going to talk about today are exclusively focused on bees, mainly bumblebees and honeybees, and predatory insects, insects that aid in biological control. Wasps, lacewings, and ladybirds. So yes, some non-eusocial species have snuck into this talk, which I hope you'll forgive me for. Likewise, I think most people will be aware of neonicotinoids, and that's because they are the most commonly used insecticide in the world. And they're the most commonly used insecticide in the world for two main reasons. The first being that they're systemic. They can be used as a seed treatment, as a foliar spray. And because they're systemic, it means the entire crop is protected from pests, which is highly advantageous if you're a farmer. And also they specifically target the insect's nervous system, which reduces the threat to vertebrate wildlife. Now, because these insecticides are systemic, they also get into the nectar and pollen of treated crops and surrounding wildflowers and weeds, where they can have significant sublethal impacts on beneficial insects, and these effects are widely documented. Now, this evidence has led to neonicotinoids being restricted in the EU. 4 commonly used neonicotinoids are now banned from agricultural use in the EU. But regardless of this, neonicotinoids are getting less effective at their job. The number of pest species which are resistant to neonicotinoids is steadily on the rise. Now, this has led myself and others to begin to question what will replace neonicotinoids. We haven't really changed how we farm. We are still heavily reliant on insecticides. So what will come in that will replace them, and do they have similar effects on beneficial insects? So I'm going to talk about 2 insecticides today. The first is this one. Now, I'm not even going to try and say this because I would just end up embarrassing myself. One of my former PhD supervisors, Mark Brown, informed me that his group are calling this Fluffy. So I'll probably just adopt that name for the rest of the talk. And the other insecticide I'm going to talk about is sulfoxaflor, which is the insecticide on which my PhD was focused on. Now, what's important to know about these 2 insecticides is that they have a similar mode of action to neonicotinoids. They work on the insect's nervous system, but they're chemically distinct. They're classed in different chemical groups. Now, this is important for marketing reasons, obviously, but it's also important because there's evidence to suggest that these insecticides are effective at targeting pest species which are resistant to neonicotinoids, which of course is very important if you're a farmer. So as I alluded to, I first became interested in this during my PhD, where I did an experiment to see whether sulfoxaflor, this novel insecticide, influenced bumblebees. I reared up bumblebee colonies from wild-caught queens and monitored them throughout their life cycle. And what I found was that there was a 54% reduction in the number of males and queens produced by colonies which had been exposed to a field-realistic dose of sulfoxaflor, this novel insecticide. Now, of course, if you were to scale that up to an entire population, you would expect that these effects could have severe population-level consequences. Now, whilst I hope that that was an important study, and I hope that it will kind of get more people looking at these insecticides, it is just one study. So, around May, when we all had to think of different things to do, one of the things that we decided to do was to synthesize all the available literature on novel insecticides and these 2 novel insecticides and the potential impact on beneficial insects. And to do that, we did a thing called a meta-analysis. So for anybody who isn't aware of what meta-analysis are, it's where you effectively go back through the literature and you extract as much data as possible. And then from that, you can put all those data sets together and try and get some sort of nice, robust, juicy analysis to really try and understand the potential impacts. So I started off by doing a literature search of 8,000 papers, which was fun. And from those 8,000 papers, I whittled it down to 26 papers, which were included in a review that I've written, which is linked at the end of the paper, at the end of this talk. From those 26 papers, we were able to extract data from 19 of those papers, which resulted in 98 effect sizes across the 2 insecticides. And this is what we found. So here are the results, which I haven't filled in yet, of the first insecticide and the potential impact on beneficial insects. So this is a forest plot. I hope you can see my mouse, but here we have, at the bottom, we have Hedges D values, which is effectively the overall effect. Now, if the overall effect is on the -1 side, or the minus side of this graph, then we would suggest, then we would predict that the overall effect of the insecticide on beneficial insects is negative. If it's on the plus side, then we would suggest that it was positive. And if the confidence intervals cross the 0 line, then that would suggest that there's no difference between the control and treatment insects. So I'm just going to go ahead and fill in the first result. So if you look down here, this is, if we include every effect size that we were able to gather from this insecticide, you can see that there's an overall negative effect of the insecticide on beneficial insects. If we break that down into the 3 different data categories that we collected, mortality data, which is fairly self-explanatory, fitness data, things as reproductive output or ovary development, or behavioral data, foraging, cognition, you can see that in all 3 of these data categories, there was a negative overall effect of the insecticide on beneficial insects. Again, if you were to break that down into field realistic or above field realistic dosages, we can see here that the overall effect at field realistic dosages is negative. And we can see that that effect is consistent across pollinators and predatory insects, but probably need to err on that, be a little bit cautious interpreting the predatory insect results because the sample size was low. Overall, quick summary of this slide would be that we found an overall negative effect of this insecticide on beneficial insects. Here is a very similar plot for sulfoxaflor, which is the other insecticide we looked at. You can see that the sample size of this insecticide is much bigger, and we can see there's an overall negative effect again, and that effect appears to be much stronger. If we break the data down into 3 categories, we can see that for the mortality and fitness data, there are overall negative effects, but there wasn't an overall effect for the behavioral data. But perhaps most importantly, we found that these effects were consistent at field realistic dosages. And again, these effects were consistent across pollinators and predatory insects, and the predatory insect sample size is much higher than it was for the first insecticide. Now, I've probably gone through that quite quickly because I'm only have a short period of time, but the overall takeaway message from those 2 slides is that novel insecticides appear to have negative sublethal effects on beneficial insects, and whilst we don't directly compare them to neonicotinoids, they appear to be quite similar. Now, what this really demonstrates, in my opinion, is that the regulatory process by which insecticides are licensed or not licensed doesn't appear to be protecting beneficial insects from the unwanted consequences of insecticide exposure. So we've made a couple of suggestions within our review paper. The first being that sublethal assessments of native bees should be mandatory within the regulatory process. We believe that a broader taxonomic group of insects should be assessed within the regulatory process, including things like wasps and predatory insects, important predatory insects. And it's increasingly important that the interactions between agrochemicals and other stressors should be included pre and post licensing. Also, I've written an op-ed in an attempt to crack America about the regulatory process over here in the States, which you can read at that link. Also, you can find more information about the review that we did at this paper here. Last thing is to just thank you guys for listening, thank the organisers for putting this conference together, thank my postdoctoral mentor, Felicity Muth, and everybody in the lab for just being awesome, and I'll happily answer any questions.